Welcome back to my Bioinformatics practical class. In this video, viewers will be able to understand what is NCBI, search for a gene or sequence, download DNA sequence in FASTA format, save sequence for used in Mega or other bioinformatics tools. So, what is NCBI? NCBI stands for National Center for Biotechnology Information. NCBI is one of the world's largest biological databases, and it provides access to bioinformatics tools such as DNA sequence, RNA sequence, protein sequence, scientific literature, and genomes. Why do we download sequence? Why? Biological sequences are required for sequence alignment that has to do with comparing two or more sequences for similarities. They are required for phylogenetic tree construction, that is to determine the evolutionary relationship. They are also required for mutation analysis, identifying changes in genetic sequence, and for comparative genomics, that is analyzing differences between species and their genomes. Then they are also required for evolutionary studies, that is understanding mechanisms of evolution. So let's move to a practical session where we download DNA sequence from NCBI in FASTA format. Go to your Chrome browser, open your Chrome. As you can see, it has opened the NCBI library. This is where you can search for your DNA sequence or protein sequence. Click the left side where you have all databases and select nucleotide. If you want to download a protein sequence, you are going to select protein. There is a place for protein. This is protein, but we are downloading DNA sequence, so that is why we choose nucleotide. Then on the search bar here, you can type in the DNA sequence you want to download. It could be the gene of anything. It could be the CO1 gene. It could be SOD gene. Any gene of your choice that you want to download the DNA sequence, you can type it here. So for the sake of this example, let's use the CO1 human gene. Then after typing it, you click on search. Okay, after typing Homo sapiens CO1 gene and we search, you can see the result. There are about 1,152 items found. You can see when you scroll down, you see many genes associated with Homo sapiens. So you can pick any one of your choice. Let's go and pick the first one on the list. The first one on the list, which is this Homo sapiens isolated MB3027 mitochondria complete genome. So click on it. Once you click on it, it will open the information, the gene information. You can see these are the gene information, the locus where it was found, the accession number, and other details, the reference number, the author, and other details. So to download this particular gene in FASTA format, you click send to here on your right hand side. At the top right hand side, you click send to, then you select file. They say choose destination, choose file. Then they say choose format. Which format? change it from GenBank to FASTA. Change it from GenBank to FASTA, then click create file. It has downloaded sequence.fasta. It has downloaded. It's now in your system, your download folder in your system. You are going to see this is the sequence we have downloaded. So this sequence can be used for analysis, for alignment, and for other analysis in Mega or other bioinformatics tools.